Dr. Rainey runs a successful soybean breeding program at Purdue University and has released 8 soybean varieties. She holds 3 plant variety patents, has 2 patents granted, and has over 50 research publications. Dr. Rainey has given dozens of invited lectures on the breeding, genetics, quality, phenomics, agronomy, physiology, and pathology of soybean, and she teaches introductory to advanced topics in genetics and crop improvement at Purdue University. So with that, I will hand it over to you, Dr. Rainey, for you to showcase your soybean breeding program.
Sure. Thank you so much. And thanks to the committee for inviting me to do that. And as I was explaining earlier, I was stuck in Iowa last night because of weather. And so I'm actually in Culver's, Illinois, to be able to deliver this, which is why you might see people walking by behind me with the French fries and things. But I'm happy to talk about soybean breeding. I think it's important for any TV to highlight what plant breeding is and looks like. And I thought I would start out probably with a good chunk of this presentation talking just generally about soybean breeding, and sort of starting with the history, which I'm showing on this slide here. So when I was in Iowa, a colleague from University of Nebraska asked me if soybean breeding had largely gone the way of corn breeding and become, mostly a private sector endeavor. So I wanted to address that kind of conception, which was first that here we have yields of corn and soybeans from 1866 to 2024. And really, corn breeding really has never been much of a public sector activity. Largely the hybrid development occurred just in the private sector. And it really got its inception in the 1920s. And that was this beginning of plant breeding in the seed industry with the introduction of hybrid corn in the 1920s. And then there was a recognition that soybean could be valuable around the end of World War II. And that's when the U.S. started investing in public breeding of soybeans in the 1940s. And then soybean was originating in East Asia, of course, and was brought to the U.S. as a forage crop. And that slowly, or pretty quickly, began to be converted into an industrial crop for the protein and oil that it produces. So for the first 50 years, more or less, soybean breeding in the U.S. was entirely a public endeavor at the land grants and USDA. And actually, the varieties were publicly released and were distributed by the seed companies as sort of a service to the farmers who were buying the hybrid corn seed, and then they needed the soybean seed for rotation. And so that's how the public varieties were largely distributed. Then around the mid-1990s, we saw the introduction of the first, or the commercialization of the first GMO crops, and Roundup Ready soybeans was one of them. And that's when you really saw the private sector investment begin when soybean breeding was the end of the 1990s. But, you can see that frustratingly, the yield increases, have not followed the sort of exponential increase that corn has for soybean. So there's many reasons for that. But one takeaway I think that students should take from this is that even though we may think of corn and soybeans as being these sort of equivalent industrial crops with a lot of private sector investment, corn still has gotten vastly more investment in terms of scientist hours and money in its improvement compared to soybean. And I think that's probably still somewhat true today. And then soybeans are more difficult to breed than corn, and I'll kind of go into that a little bit. So I had a few notes here on the presentation. So as we do see an increase in genetic yield potential over the decades in soybean, those are largely attributed to increases in the reproductive development window in soybeans, so like flowering earlier and maturing later. So shifting those by a week or more over time. And then as well, we evidence from the decades of yield studies, looking at varieties released in different decades, all in the same modern environment, shows that the yield gains have largely been attributed to increasing biomass production in soybeans. So we've attained increases in yield by increasing the overall biomass production, which of course is linked to correlated with the increase in reproductive development, among other things. The other thing to note about if you're considering the history over decades of soybean breeding is that it's an inbred crop, and it's largely inbred. There's not very much diversity in soybean because it's inbred. And because we know that there were just a few so-called founders, maybe 15 to 20 accessions or lines brought from East Asia that make up a large part of the basis of North American soybean germplasm. So it's just something to recall. There's also soybean is inbred and can show large segments of linkage distinct from for example, across chromosomes that I'd even heard rumors over the years that some private companies had chromosomes entirely fixed with no variation on them in soybean. That was sort of an old wives' tale, maybe, or an urban legend about soybean breeding. Okay, so moving on, this is a familiar image, probably so many of you, that's put out by Economic Research Service, that just shows or emphasizes that herbicide-tolerant GMO soybeans were quickly adopted, and the majority of acres in soybeans have herbicide tolerance. And I'm not going to go into all of those traits. It's very complicated, and that's a large part of breeding soybeans in the private sector is the trait integration or introducing new forms of resistance to herbicides and stacking them, et cetera, and rolling all of those out. And so as I move on, I just want to mention I'm not watching the chat. I think I would probably get distracted. So we'll just sort of hold questions till the end, I think. So just to provide some context for the scale of soybean production and the breeding that supports it, in 2024, there are 86.1 million acres in the U.S. And it's in any given, and that fluctuates a little bit, but that would be probably a good average. And from what I understand, it's pretty much maxing out available acres that could be put into soybean production, when you consider rotating with corn, if there aren't a lot of additional acres to take over. Though, of course, there's been a very good success story in soybean breeding of expanding the range, north, south, east, and west, essentially, but definitely into North Dakota and higher latitudes as a result of plant breeding. And in general, soybeans in the United States are worth anywhere from $15 to $20 billion annually. So one major feature of soybean breeding is a so-called industrial crop, where the seed is valued for its component composition and it's not largely consumed by humans. And so the seed tends to be around 20% oil, and then the seed is taken to elevators for the crush, and the oil is separated from the meal, and the meal has a large portion of protein content that's valued for animal feed. And of course, you may have heard that you can consider chicken reconstituted soy protein. So the biggest uses of soybean are for oil and protein, and the vegetable oil in the grocery store is, of course, soybean oil. And then a new use that's really exciting of soybean is for sustainable aviation fuel. And that is a big driver in demand that may really change the economics of soybean as well as the goals for breeding. The sustainable aviation fuel is made of a number of oil sources, but the soybean being sort of the primary constituent of the sustainable aviation fuel. And then I just want to take a moment to say, having been a soybean breeder for almost 20 years, that all of these seed composition components will sort of shift or change in their priority over the years, and it does make it challenging for soybean breeders. Not too long ago, we had plant-based protein was a massive focus in the U.S. and there was the thought that we needed to, work to increase protein, and then it quickly shifted to sustainable aviation fuel, and oh no, we want to work to increase oil, with of course the relationships between those being antagonistic, and so just to point out that soybean breeders kind of feel like we're always chasing one composition profile or another, and it can change pretty quickly depending on things like trade wars and policies on food and fuel, etc. But it is a very remarkable crop in terms of the amount of oil and protein it produces. If you think back to the first slide with the yield versus soybean, the corn versus soybean yield chart, the yield potential of soybean is so much lower than corn, partly attributable to the fact that the oil and protein are metabolically expensive to produce. And so it doesn't, you don't expect to ever close that yield gap that makes you with corn. So I just, again, this is for students. I wanted to kind of present some general concepts around soybean breeding. So of course we see that protein and yield potential are negatively correlated. So as we increase yields, we may see reductions in protein, or if we increase protein composition, we'll see a yield penalty. And that relationship is pretty hard to break. And a lot of investment has been made in trying to identify QTL that may increase protein without affecting yield per se. And, there's definitely room for maybe incremental improvement there. And then, of course, we see a negative correlation between oil and protein. These are all just these trade-offs that exist. As you know, in plant breeding, you can't improve everything. You have to set your objectives, because you get trade-offs as you push these things forward. And I think it's interesting to just note that I'm on the advisory committee for a lentil breeding project in Canada, and some of the field data some of the data from the field that I saw there did not show a tight correlation between protein and yield in lentils. And I thought that was evidence that there was a lot of room to improve yield, probably. So I think these correlations are expected. And if you have a grain legume that doesn't have a tight correlation between protein and yield, then that's evidence that their yields can be improved. Another aspect of soybean breeding to consider, not that we do much to improve it, is that, of course, soybeans are legumes, and so they fix nitrogen. And, there isn't a lot of course, it's very expensive to improve the unseen root traits. And so you don't see a lot of breeding focused on it, but it does play into these relationships that you see between protein and oil and protein and yield. And with the desire, I mentioned that there were 80 plus million acres of soybeans, and that the oil from them can be used for sustainable aviation fuel, and they're to meet the projected demand for soybean oil for sustainable aviation fuel, we would need to produce something like 20 million more acres of soybeans, which we can't do. And so it does bring into focus then the role of the nitrogen fixation, which is positive for providing a biological source of nitrogen for both corn and soybean production. But if we were to reduce nitrogen fixation, we could potentially get higher yields of oil. And so this is some of the things that breeders are talking about and agronomists right now is that kind of relationship between yield, oil, and nitrogen fixation. So this is a figure that might be familiar to people. These colorful latitudinal zones represent the maturity groups, the so-called maturity groups in soybean. These are a classification system that soybean breeders use to describe the days to maturity in soybean, and they accommodate several days of, like any given maturity group you see here, like a group 2 or a group 3 would accommodate several days of differences in maturity. And when you think back to the lack of the sort of slower pace of improvement of genetic yield potential in soybean, consider that this system really introduces a lot of complexity into soybean breeding, and it makes it difficult to move traits across germplasm from different regions. The relative maturity groups are controlled by so-called E genes. Several of them have been described. I think there are several more left to be discovered. I think a take home about E genes is that, first of all, there are a few major ones, E1, E2, E3, that have been discovered, but the allelic effects of them maybe haven't been defined in modern environments. So the genes may have been discovered in the 1970s and described in germplasm and environment and management systems that existed then. And it's been hard to collectively update the effects of E genes. And then the E genes make it hard to sometimes predict by a genomic prediction or just reading what your maturity will be to predict the maturity of wines that you're selecting. And as well, the maturity data are very important to collect and require many environmental data to really accurately describe the relative maturity of a potential variety or line. So take home from this is that, and also soybean is the most saline sensitive crop in the world. It's considered the most saline sensitive crop in the world. These E genes are interacting with photoperiod, planting date, etc. So this is one of the things that complicates soybean breeding. And as well, I had been told by a private sector breeder that I will go unnamed, I guess, that if the traits, the GMO, like herbicide tolerance or other types of traits were to somehow go away tomorrow, let's say through an executive order, who knows, but if they were to go away, not that we want them to, that maybe the public sector breeders could compete with the private sector in terms of control per se, because there's a lot of genotype by environment interaction created by the interactions of these E genes, and the public soybean breeders tend to focus on smaller regions of adaptation. So just something to consider. So I wanted to talk about, we have these stages of development in soybean, and we tend to see a very tight correlation between maturity, date maturity, which is described as R8, and yield. This is something as a breeder that I want to see. To me, it tends to be indicative that I have both good yield data and good maturity data when I see that tight correlation. And then in a year where you might have a drought or something like that causes the maturity to kind of deviate from this tight correlation, it's just a good indicator. And then also, one of our targets for improving yield in soybean is to take the R1 and R8 dates and sort of expand them to lengthen reproductive development, or to basically, shift it the way we want it to go. And I really try to advocate for funding to discover more flowering maturity genes that I know are there, so that we can dial in flowering maturity for our specific environments. Right, I thought I had I think that's probably the bulk of what I wanted to say here. Then, one other thing is that the R8 score is the day that 95% of pods are brown, and that has traditionally been collected visually, and then it's been a use case for drone phenotyping, and I'll talk about that a little bit later. Just wanted to point that out before moving on. So I had mentioned that there are many public soybean breeders. So going back to the 1940s, the U.S. made investments through land grant system as well as through USDA and soybean breeding. So anywhere where you can imagine soybeans are cultivated, there's probably a public soybean breeder at Land Grant University in the United States. So there's about 16 or 17 of us at the land grants. And we are largely supported by checkoff dollars, which come through states, the national and the regional funds, et cetera. So the public soybean breeders do tend to collaborate quite a bit, as well as every one of these breeders would have a functional public breeding pipeline in their states. And I forgot to actually put a star on Indiana, so there's one in Indiana as well. And as evidence for this kind of history of collaboration, we have what's called the uniform tests. They are cooperative yield trials that are organized by the USDA, where all of the breeders, contribute our advanced lines and then grow the tests, et cetera. And in 2023, there were 100 locations of testing for the uniform test just in the North Central region alone. So it exists at a large scale that I think is probably unprecedented amongst public breeding and other crops. I really want to emphasize that the public soybean breeders are validating the performance of their selections against everyone else's in a very robust multi-regional trial. And it really enforces having agronomy and breeding at a certain scale and keeps us sort of accountable for what we do, right? So we're not sort of breeding in a vacuum of validation of what we're achieving. And it's a great resource to build on. In fact, a publication just came out from Aaron Lorenz, where he was finally able to sort of incorporate all of this data going back to 1941 into our database to allow it to be sort of analyzed outside of its traditional uses as just providing performance data for releasing lines, et cetera. Another thing to mention is that since all the public breeders are participating in our multi-environment trial, it sort of predicates that we then exchange germ plots with each other for crossing. So that's nice as well. And I think that it emphasizes that public breeding for soybean is strong and is occurring at a scale that's not equivalent to the private sector, but at a scale that I guess you would see in the private sector potentially. Though, of course, when I talk to breeding organizations, we may have in the public sector tens of thousands of plots, and a large company might have 1 million or 1.2 million. Of course, that would be across all the breeding pipelines. These cooperative trials are just for like preliminary and advanced trials after just 1 or 2 years of field testing. Okay, so one of the biggest challenges of soybean breeding is we don't get very much seed, and it's very hard to make crosses. That dictates all of how soybean breeding is accomplished, and that's why we don't have hybrids and everything else. So soybean flowers are quite small and easy to damage, difficult to emasculate, etc. So you get a very low success rate on crossing. And then if you do make a cross, you get 1 or 2 seeds, and the fact that you may only get 1 or 2 seeds per cross, and then the seed, each pod only produces 3 seeds on average in soybean, just means that it's very difficult to get enough seed in the breeding process to start making selections and getting valid replicated yield data. It's just a challenge that exists. And it's a point to just about. So you're geared towards students. You see all these flowers. Grain legumes are notorious for producing lots of reproductive organs and shedding them if there's any kind of stress. So soybean is considered taking escaping stress and will yield under conditions where where corn may not. So, for example, if you have a drought at the wrong time in corn, you're going to see a severe yield penalty. If you get a drought during reproductive development of soybean, it will just kind of tolerate that and shed these sort of inexpensive flowers and then set seeds when the conditions are favorable. So that's one of the reasons why expanding your window of reproductive development has been a successful strategy of attempting higher yields, just to give more time in that window. Single seed descent. This is actually scanned from my textbook in plant breeding from graduate school. So soybeans are largely bred through the single seed descent method, which of course was actually invented by a soybean breeder for USDA in the 60s. So conventionally, or I guess until recently, or until the past maybe decade or two in the private sector, this would be how soybeans were bred, and it may be this is largely how I manage my breeding program as well. So single seed descent will be target to create a fairly large F2 population, and you'd save one seed in theory from each plant for a few generations until you've achieved sufficient inbreeding. Then you go in and, using visual selection, take a subset of your inbred plants. Typically, most soybean breeders are doing this actually at the F4 stage. You get a head row or progeny rows coming out of our individual plant selection. And then you go into sort of the replicated testing. So a couple of notes about that. We don't usually actually save one seed per plant. You might do something called grab sampling, where you kind of try to sample these early generation populations, each plant sort of equally, with a few seeds or a few pods from each plant. And of course, one of the advantages of a single seed to set method is that you separate your inbreeding from selection, and you could inbreed to get a stable line before going into replicated testing, and do all of that outside of your target environment, doing it in layer nursery, etc. And this is a relatively inexpensive way to breed soybean compared to the early generation testing that the private sector does. This is a slide that I received from Trizilla at Corteva when I asked him to present what may be more typical in the private sector, which involves early generation testing. So lines are actually established in the private sector, usually in an early generation stage, and testing is performed in early generations, and then there's sort of a cleanup or purification or resampling process that happens once the most promising kind of families or lineages are identified, which is I call that that and some other practices may be a more expensive way to breed soybeans, but of course that's how you're going to get more rapid genetic gains, essentially. So, as I understand it, for example, at Corteva, they take advantage of this method of single seed descent by doing the crosses in the early generation management, all in Puerto Rico in the off season, or not in the off season, but just outside of the target environment, so that they can basically have people who are sort of expert at crossing and managing early generation soybeans, doing that year round, and get multiple generations per year that way, which of course, the easiest or the most impactful way to increase genetic gain in the low breeding program is to reduce the cycle time by obtaining multiple generations per year. And so it's amazing to me that what the breeders at a company like Corteva get back from a crossing and population management operation like this would be, I think, several dozens of environments of testing in the first round of testing. Whereas for newly, I might only have 1 or 2 environments for progeny rows and 2 or 3 for the preliminary yield trial, et cetera. So if you can devote the funds to it, you can get quite a number of, you can kind of maximize seed production, et cetera. And of course, genomic prediction can be applied anywhere throughout the process of soybean breeding. You could do it at the F2 stage. You could do it when you're selecting your individual plants. You could apply it to progeny rows. And as I understand it, it's applied at multiple stages throughout the sort of inbreeding and selections in the private sector breeding. And then there's Charlie highlights a couple of things here for what Corteva might do, including that at some point once the advanced lines have been selected, there is a whole process of trait integration, which refers to, of course, introducing the traits for herbicide tolerance and other things. And Corteva has invested quite a bit in drone phenotyping, and all of that information can go back into building the selection models in the earlier generation stages. As we talk about drone phenotyping of soybean, it's worth noting that Corteva did sort of pioneer in the application of drone phenotyping to measuring soybean maturity, and that this has been one of the earliest and most persistent and valuable applications of drone phenotyping, I think, in breeding of any crop is the soybean maturity. So traditionally, without the use of drones, visual notes have to be taken in the field, 3 times a week on the day that to describe the day that 95% of pods are brown, which this has been converted by most breeders into a drone-based regression prediction of the day of maturity that's based off of color. And this is all predicting the drone-based color estimations are predicting R8, which itself is a prediction of physiological maturity. That's just something I like to point out. So we're predicting something that predicts something else when we use the drone metrics, which is just fine. It's also worth noting. And this is the original publication describing how to do the visual estimation of maturity. I do work a lot on drone phenotyping, how to use drones to predict yields and other traits in soybean. And I like to emphasize that to measure color, you just need sort of, this just requires sort of blurry blobs of color from the drone, and that it takes, which is relatively easy to do. And then measuring other traits may require a lot more sort of precision or 3D modeling and other tools. Okay, so this is a great application of drones. It's also a very sort of low-hanging fruit. It's relatively easy compared to other things you might try to do with drones. So this leads to another thought that I had, that, when we get into more of what I've been working on recently, to remind everyone that historically, row crop breeders have relied on measures of yield from combines, which really weren't widely available until the 1970s. And then what we observed visually to select superior lines, and now drones enable us to measure new traits that we maybe weren't able to measure previously or intuit even existed. So that's one of the things that motivates what I do. I have a history of working with drones that goes back to, in my breeding program, that goes back to around 2012, 2013. And these are sort of older information, but I just wanted to describe the progression. In the nested soy NAM, or Nested Association mapping population that was planted about 10 years ago now or more, we started flying drones and measured canopy coverage of the traits. That here's sort of a picture of what a canopy coverage is. It would be percentage of pixels that were canopy. It becomes a longitudinal trait that changes over time, so we have distributions of canopy coverage over time. And this is a presentation of a GWAS, or where they'll, instead of using peaks to represent significance of association of regions of the genome with the trait, we now have a color scale and then some sort of measured time on the y-axis. It gives you a representation of how a trait like canopy coverage that's continuously variable would have different genes controlling it throughout development. And this is something that I've built on quite a bit. When we published a paper about this in 2017, that we had found a large QTL that confirmed canopy coverage as well as impacting yield. And that was confirmed subsequently by all of the yield-based mapping of the soybean population. It's just something I've always liked to point out to people, that it took 3 environments of drone data to find this QTL and 17 environments of yield data to find the same QTL. And so I always been a proponent of using RGB, just regular red, green, blue cameras in drone phenotyping in crops, because it will identify sort of the low-hanging fruit or the large effect genes, or the traits with a big signal. also recall that soybeans fix nitrogen, which I showed you with nodules on their roots earlier. So what that means is that soybeans tend to be uniformly green, and there's not a lot of color variation because it doesn't lend itself towards spectral indices, etc. The canopy coverage QTL we discovered, have been validated multiple times in other studies, and we learned that measuring the canopy was the canopy traits do have a high correlation to yield potential. So I mean, particularly on sort of a population like an early generation population where there's a lot of genetic variance for yield potential, you're going to see a tight genetic correlation of canopy traits with yield potential. Once several rounds of selection have occurred and there's not a lot of variation for yield, then there may not be canopy traits a lot as predicted. So one of the things that I focused on subsequent to kind of my early work using canopy features and mapping them is to try to to exploit that genetic correlation in the breeding pipeline. And so this is just similar. It's a recapitulation of the single seed descent kind of method of breeding. But when we get to this progeny row stage where we don't have very much seed, we have very little seed where, like I said, we can maybe just do one short row or two short rows in one or two environments. So it provides low quality yield data for selection. So when I started, I'd say the first half of my career as a soybean breeder, I was doing visual selection of soybean progeny rows, and that was definitely the state of the art then. And so we did a number of experiments that showed when we used drone data, we could pick a higher yielding lines than if we didn't. And so I was going to show you a few results from that. When we do a selection experiment, you need to use multiple sources of information to select your lines. And you can come up with as many selection categories as you want, but you have to make sure that you're using the same selection intensity for each selection category. And one of the things that I've also learned doing selection experiments is that you then need a random selection category as a control. In the animal literature, selection experiments are common and they use a negative selection as a control. But there's a lot of genetic reasons why a line might be performing badly in a field. So this is representation of what it looks like when we like a Venn diagram of different selection categories, and you expect to always see overlap between them. So if you're using yield as a source of information for selected, and then you add like yield and drone data or a genomic model, et cetera, of course they're going to pick many of the same lines, but as they're different ones. Sure. So we did this in our own breeding pipeline. This was published a while back. And we can see our advanced field trials from the early and late maturing classes for 2 years. And these are the selection categories or sources of information that selected the 10 highest ranking lines in our advanced field trials. So these 2 stages after the progeny row selection phase. And you can see that anything in red would not have been selected without drone data. And so it was using canopy coverage or yield adjusted for canopy coverage as selection criteria has been adopted by a number of public sector breeders, and I hear it's also used in the private sector. We repeated this across 4 breeding programs. As I mentioned, the public sector breeders collaborate quite a bit with support from farmer dollars, and we sort of saw the same result across multiple breeding programs, more or less. Basically, anytime we use another source of, the more sources of information we use for selection in this this unreplicated sort of early generation progeny row phase, the more likely we are to select high yield lines. I don't have a figure for it, but another thing that we've learned from doing this is that using sources like drone data often helps us to pick lines that are earlier per se. So soybean breeders have to be careful to if we do not account for maturity in selecting on yield, we will only just select we'll select just for later maturing soybeans. you have to try to select on yield per se by adjusting your yield data for maturity date. And so we found that using drone data tends to select lighter, higher yielding per se, and not just because they're lighter. I did some work to commercialize the image processing pipelines that we developed several years ago with a product called Phonomics that was acquired by a seed company about a year ago. And it was based on a principle that we call algorithmic plot clipping, where you use algorithms to extract your plot images out of every raw image collected by a drone. And this is a departure from what most people do, or what a lot of publicly available image processing pipelines involve, generating orthomosaics and then clipping like the single kind of blended or average image from orthomosaics. And it's an important point that I wanted to make. I've presented this at an APV this summer. That algorithmic plot clipping is what gives us the scalability or precision that's needed to implement more regular drone phenotyping throughout all stages of breeding pipelines. And there still aren't a lot of tools existing for that. So this algorithmic plot clipping was developed based on because we did have large scale yield trials in the public soybean breeding endeavor. And that's just something I want to emphasize. The farmers are supporting the public soybean breeders in all these states, and then we collaborate and put out these large scale trials that are, look like something that you might see in the private sector or at a major scale. And then that drives this kind of innovation to the point where, necessitated creating a efficient tool that was then recognized as being valuable by the private sector. So it's just something I would emphasize about how public soybean breeding works. And I think I've said it before, I don't find a lot of utility in spectral data from soybean because it fixes nitrogen. And I just am really more focused on what we can measure throughout development with RGP. I'm just going to move on because I see that I'm running out of time. We've published some papers on yield prediction. Of course, if you're in a breeding pipeline, there are, you can one way to increase genetic gain in a breeding pipeline is to increase your genetic variance, basically have larger, bigger, or more bigger populations. And one way to achieve that is by using drone-based predictions of yield. Definitely the bottleneck to implementing more yield trials, for example, is the expense and time it takes to actually harvest spots to measure yield. So if you can predict it with drones, that will be beneficial, and that's something I really see a lot of interest from in the private sector is, can we predict yield in more advanced trials and with drones and use that for increasing gain as well as risk mitigation, et cetera. I think we've all observed, or I've been around long enough now to observe, that the environments that we experience in the field are getting more extreme and stressful. And I think we're more likely moving forward to see more of that, and that you're more likely to experience a loss or a failure or an inability to harvest your yield trial for whatever reason. And I think routinely implementing yield prediction would help with that as well. So that's something I've been working on with all the public soybean breeders. These are 3D point clouds of just 6 public breeding lines where biomass samples were taken for the purpose of developing biomass prediction capabilities from drone imagery, and for use in predicting yield, whether it's incorporating phenomic and genomic data or just from the drone alone. And you can see there's actually a lot of information from 3D modeling of RGB images to be extracted. And I currently have a postdoc working on that. So one of our collaborative efforts across multiple soybean breeders has been the Soygen project, which is funded by one of the checkout regional funds. And one of our current activities that's the big focus for me is a yield trial designed by the PI Aaron Lorenz to develop genomic prediction models for predicting G by E when phenotype by environment interactions in soybeans, to be able to sort of predict where soybean might be adapted, even if you don't have testing data for it. And my lab is receiving all of the drone data from the collaborators growing these panels of soybeans. All of the drone data is sent to my lab, where we're creating 3D models of the canopies of the soybean plots to collect sort of longitudinal structural features and canopy traits, and look at how that predicts yield within the genomic prediction framework as well as per se. So that's one of the big efforts of my lab right now. And so we've been working on always improving. There's never enough ground truthing data for canopy, for biomass production and other canopy features. This is some kind of preliminary hot off the presses data that we have from trying to ground truth biomass and develop the ability to predict it. Recall that biomass production, that increase in biomass production is what's driven the yield gains over the decades in soybean breeding. And we know that actually the harvest index of the wheat varieties has approached the theoretical maximum. So further yield gains could come from increase in biomass. And in biomass, there's a good kind of summary trait for many structural features, as well as the soybean component and calculating the physiological efficiencies, which is one like radiation use efficiency, water use efficiency. That's one of the reasons we focused for a few years on it. I do wish there was more funding for ground truthing biomass, and I have this vision that sharing ground truthing biomass data and the associated imagery could be a real motivator for everyone to contribute to like a general pool of data, which you could develop prediction models based on your goals if you're a breeder, agronomist, etc. Another application of one of the other kind of areas I'm looking at right now in breeding is on resilience, herbicide resilience. So this is a picture of the John Deere See and Spray technology that many of you may have heard about. It's exciting and just now being deployed widely, and that's using sensors on a spray bin to detect weeds and then sort of inject or having a sort of a spray of concentrated herbicide on just the weed itself. So it reduces herbicide use and helps to mitigate resistance to herbicides, because you can deliver a more concentrated dose to the weed. But for See and Spray to be effective, you need a pretty low weed population in a field. If you get a lot of weeds, you might as well just spray the whole field, basically. And so we're seeing that soybeans are going to always receive higher doses of what's called pre-emergent herbicides that are put on the field after planting. And there really isn't resistance to pre-emergent herbicides in soybean. They do damage soybeans, and it's just kind of a spectrum of resilience. So I've been looking at using drones to quantify resilience to pre-emergent herbicides, because the state of the art in weed science is to do just visual scoring of these of the relative resilience of different lines. And so we have implemented field screening protocols where you can screen thousands of lines. Those could be breeding lines, mutants, trait formants, whatever, germplasm, whatever you want. This provides a high throughput method of assessing resilience to herbicides, and you just distribute. It was actually a field map. You distribute your resilience sensitive controls throughout the field. They're visually scored by a weed scientist. You collect drone data on all of it. You can develop a regression equation that will predict the relative resilience of lines based off of things like color, canopy coverage, and biomass, growth rate, etc. And that seems to have worked pretty well in terms of being repeatable across years. And we have a greenhouse screen going on right now to confirm the observations we've had in the field, and that will validate this method, and hopefully we can begin screening something like transformants or mutants, as well as trying these methods with other classes of herbicides. Since that's so important in soybean, herbicide resilience and resistance generally. So with that, I'll take some questions from the group.
Thanks, Katie. That was a great presentation. If anybody has any questions, feel free to either raise your hand and put them in the chat. We have a few from online too that we can start out with if anybody doesn't have any questions right away. Now, I personally had a question. You presented on predicting yield. And especially in years where maybe you get a storm and you aren't going to harvest it. How do some of those studies deal with the G by E that you usually get with yields or with multi-year?
It's actually hard to. We haven't been able to address that, right? So we've been able to predict yield in like a single environment or a few environments, and we're sort of laboriously building up the multi-environment data set of longitudinal drone data and yield data that you would need to address that question that you ask. And you're right, and I mean, I think that is the emerging question in breeding in general, is how to take advantage of predictive analytics with variable weather and G by E and things like that. And so, I hope that the Soygen data set that I know that the Soygen data set I showed you, we've had 20 environments in 2024. We'll have another 20 in 2045. That begins to be at the scale that you need to answer those type of questions. How well can you predict yield, or what kind of information are you going to need to do that and accommodate for environmental effects? And of course, it depends on the context of the, where in the breeding pipeline you are as well. Are you predicting yield early on or in advanced trials? Yeah.
And, the work you've done or you were citing, was that mostly just RGB, or were they focusing on the other,
I've pretty much worked exclusively. Yeah, I pretty much worked exclusively with RGB, and so we used canopy coverage and then structural features from 3D modeling. So the primary one being height, but we're also doing other structural features as well. So the combination of canopy coverage and height gets you pretty close to biomass in soybean. But again, this is on a limited number of environments, and that's why soybean is going to be so useful to test those prediction algorithms just for biomass alone and the ability of these canopy features to predict yield. Yeah. So when you talk about predicting yield, you do have to differentiate between exploiting genetic correlation when you have a high phenotypic variance for yield versus predicting the yield of a yield trial, itself, right? Yes. But yeah, I've focused just on RGB. Again, I don't think we've collected plenty of multispectral data, and I've yet to see a case in my own limited set, but having worked with it for several years in a few environments, I haven't seen a case where the multispectral data really adds anything to our ability to predict biomass or yield in soybean. And it's an important message to deliver, though, because spectral indices are expensive and complicated and have, that don't have the resolution that RGB does. And I'm a big proponent of having more drones in the landscape, collecting more data because they're robust and inexpensive, like we have with RGB platforms.
Yeah, Dan or Don.
Quick question, Katie. What's being done to address the limited genetic variability in soybeans going forward? How is it being expanded?
So there have been, of course, we had a curator of the germplasm collection for years that did try to do that and release lines that were breeding lines that were a certain percentage accession in the background, and there has been evidence that has contributed sort of yield genes that or enhanced yield potential in soybean. Other efforts would be, looking for these sort of genomic regions that might be valuable to exploiting germplasm. And I think, I can speak for public breeders and saying we all tend to try to, emphasize crossing to lines that have accessions in the pedigree. But I think it's just a constant battle, or a constant challenge in soybean breeding to expand diversity. Yeah.
If that's the bottleneck, though, isn't that really needing some bigger emphasis to address it?
And the bottleneck, you think you think that's what's limiting our yield, our progress on genetic yield potential?
Well, at the beginning, you kind of indicated that there were only 15 or 16 progenitor lines that make up the elite germplasm in the U.S.
Well, over the course of the decades, 60, 80 years, yes. But that was, that knowledge, we've had that knowledge for about 25 years, and there are efforts to try to extend that. I don't know. I'm not up with what everyone else is doing at the moment in terms of diversity. So, but you're right. That is one way to address it.
Are there close relatives that would make sense to exploit?
We have glycine soja, which is a very different beast from glycine max. Glycine soja tends to have a prostrate growth habit, so it grows on the ground. It's kind of somewhat perennial, has tiny little black seeds, and just brings along a lot of linkage drag. And so there are geneticists working on identifying things like shattering loci and stem terminate, all of these sort of structural features that tend to be like fixed in the cultivated germplasm, identifying where those are and trying to bring in diversity from soja. But that is a very long-term game, right? To use a wild relative, but it is possible, and there are some studies that have.
What about the potential for enhanced photosynthesis and things like that? I do see a lot of people proposing to, enhance photosynthesis through gene editing and or through just being able to select on it. I don't know. I think that the verdict is out there on whether that will be, how successful it could be to improve those. But it seems like the environment may be changing faster than the diversity of soybeans.
Oh, yeah, that's a good. I think that's a fundamental question for breeding row crops moving forward is how, there is what represents the average environment anymore. And especially when you're using predictive analytics, it does call into question what you're trying to predict if every environment starts to, the past few have been doozies. And this year 2024 wasn't so bad, but preceded by a couple of pretty wild years in terms of droughts and late frosts and all that kind of thing. I see there's one question in the chat. Getting good prediction accuracy on biomass. How would that be useful for seed yield prediction, provided the tricky correlations? What is it? I think it's. Any thoughts about that for predictive breeding? I think it's sort of TBD. It's very expensive. It's hard to get funding to create biomass prediction capabilities. So we have just limited ability to do that. And it seems to be predictable, and we're trying to deploy it now, or at least not. Every time we use canopy coverage or other canopy features in selecting early generation soybeans with a lot of variation for yield potential, we do see that it will pick higher yielding lines than other sources of information. And I didn't mention, because we haven't published it yet, but we've concluded that we get the same result in the genomic prediction. Like we've tested genomic prediction for yield versus genomic prediction for yield incorporating longitudinal biomass data, and the longitudinal biomass data is enhancing the genomic prediction of yield. And again, it is difficult to even begin to predict biomass on a large scale to answer some of the questions that you have there. We know pod counting doesn't predict yield. That in some environments that will. Soybean breeders know that there are plenty of empty pods in certain environments, and so I just think we haven't gotten to the scale yet to really test some of these potential capabilities of these technologies in many environments. Anyway. Well, it looks like.
Sorry. I was just going to say, I don't see if there's any more questions. I see exactly we're past the hour, so I really want to thank you again, Dr. Rainey, for your time, and for everyone else, thanks for listening. And
yeah, thank you for joining.
We will be putting this recording on our YouTube page as well. So thank you.
All right, thank you.
And bye, everyone.